Welcome to Lecture 21 and 22, where we will go over the process of transcription and translation. We now know that most genes code for proteins. This was first postulated by Archibald Garrett in 1909. He conducted research on how metabolic diseases were inherited from parent to offspring, and most of these metabolic diseases were due to the individual lacking a certain enzyme. Beadle and Aphrusi speculated that mutant alleles present in Drosophila were due to a defective enzyme that were defective at some point in the pigment production process, leading to the mutant condition. Beadle and Tatum came up with the one gene, one enzyme hypothesis, which states that one gene will code for one specific protein or enzyme. We now know that this is not in fact the case. I would like you to recall the central dogma theory. This states that DNA acts as a template and codes for proteins, using RNA as an intermediate. This is true for most, but not all genes. Some genes code for RNA that never ends up being translated. However, this RNA can still carry out many important roles in the cell. The central dogma theory can be divided up into two important steps. Step 1, transcription, which involves the synthesis of the RNA molecule, and step 2, translation, where this RNA molecule will be used to create a polypeptide chain, which will eventually take its conformation to form a protein. In step 1, the RNA sequence that is formed is dictated strictly by the DNA template. The overall process of transcription holds many similarities to the process of replication, such that we have a DNA strand that will serve as a template that will be copied in a complementary way to make an RNA molecule. In eukaryotic cells, this is located in the nucleus. RNA will often have to be processed into a mature form after transcription before it can be used for the translation process. This is because the eukaryotic genome is much more complex, and also because it contains many non-coding or non-important sequences. The process of translation occurs in the cytosol for both prokaryotic and eukaryotic cells. In this process, the amino acid sequence of the polypeptide chain is directly dictated by the mRNA sequence. The process of translation is carried out by the ribosome. This diagram here depicts the overall differences in the location of these two processes between prokaryotic and eukaryotic cells. Note that in prokaryotic cells, transcription and translation will occur in the cytoplasm. However, in eukaryotic cells, transcription and RNA processing will occur where the DNA is housed in the nucleus. Translation, however, will occur in the cytoplasm. We're now going to consider some fundamental differences between the two nucleic acids RNA and DNA. RNA is considered to be single-stranded, though it can form double-stranded complexes. For the majority of the time, RNA is considered to be a single-stranded molecule, so it will not form a double helix like we see in a DNA molecule. DNA is also considered to be more stable of the two, whereas RNA is considered to be labile. This means that RNA will be continuously created and degraded within the cell. There are also some structural differences between these two molecules. The base sugar in the nucleotide base will be different depending on whether you're dealing with DNA or RNA. DNA has deoxyribose as the base sugar, whereas RNA has ribose. The nucleotides present will also differ with respect to the nucleic acid. DNA has adenine, cytosine, guanine, and thymine, whereas RNA has adenine, cytosine, guanine, and uracil. There is no thymine present in an RNA molecule. There are also three different forms that RNA can take: mRNA, rRNA, and tRNA. mRNA will be important in the transcription and translation process. rRNA will be present in the ribosomes, and tRNA will be involved in translation. In eukaryotic cells, the DNA is present in the nucleus at all times, whereas the RNA is considered to be mobile. It can move from the nucleus into the cytoplasm for translation. In eukaryotic cells, we have the DNA also packaged differently. It can be wrapped around proteins called histones. RNA is considered to be naked because it does not have these histones present. Thus, it cannot be wrapped up to be condensed, like we see in a DNA molecule. We know that most genes code for proteins. Not all genes will code for proteins, but most of them do. However, there are only 20 amino acids that we have naturally occurring in the cell, and we also only have 4 nucleotide bases. This means that it must take more than one nucleotide to code for an amino acid. This is resultant because if the code only consisted of 2 nucleotides, this would mean that there would still only be 16 amino acids. We know that the genetic code for an amino acid exists in a codon, which consists of 3 nucleotides present in a row. This gives us a total of 64 possible codons present to code for 20 different amino acids. This means that the triplet code is said to be redundant because there are many different ways to code for one specific amino acid. However, this coding system is not considered to be ambiguous. This is because as long as you have a codon, it will code for a specific amino acid. This slide depicts the overall process of transcription and translation. Note that we have a DNA template strand, in this case the upper strand, which runs from 3 to 5. Also note that we have the other strand of DNA, which runs from 5 to 3. We would consider the DNA as our template strand because it is running in the right direction. Remember, in DNA replication, we had the enzyme DNA polymerase that can only read in the 3 to 5 direction. We have a similar concept that applies to transcription and translation. Also remember that this process is going to be using the complementary base sequences. This means that when a new RNA strand is produced, it is going to be produced in a complementary manner, such that it will be produced in a 5 to 3 direction, and it will also be produced by adding the complementary base sequence to the DNA template strand. Also note in the image that we have the existence of codons. These codons will determine what amino acid will rely in the polypeptide sequence. Note that the codon UGG corresponds to the amino acid tryptophan, which we see in our polypeptide chain below. For each gene, only one strand of DNA will serve as a template for the transcription. We refer to this strand as the template strand. During transcription, as stated previously, the RNA will add base pairs in a complementary sequence to the template strand. This will lead to the creation of the RNA strand. We do know that the non-template strand is also considered to be complementary. We consider it to be silent and not transcribed in the transcription and translation process. However, it is still important as it will indicate what your corresponding mRNA sequence would be. This is because both mRNA and the coding strand will both be complementary to the original DNA template strand. We refer to the non-template strand as the coding strand because it is very similar to the RNA that results from this process. However, there are some fundamental differences between the coding strand and the RNA strand. RNA would have uracil, where thymine would be present in the DNA coding strand. In essence, we can use the coding strand to read the gene sequence. This will give us an indication, once again, of what our RNA strand would be looking like. The RNA strand would then be used to translate into a functional protein. This diagram gives an overview of the process of transcription. This process can be considered a 3-phase process that begins with initiation, continues with elongation, and ends with termination. In the initiation phase, we have RNA polymerase. This is an important enzyme that will create the new RNA strand. It will bind to the promoter region. It will transcribe the DNA sequence and create the RNA transcript. Note that we have both our template strand and non-template strand. Note in this case, the template strand would be the second strand on the bottom of this sequence. That is dictated simply by the direction that these strands are running. RNA, in this case, will be running from left to right, meaning it will be, once again, reading the template strand in 3 to 5. This is because, similar to DNA polymerase, RNA polymerase can only read in the 3 to 5 direction, and it can only make a new strand in 5 to 3. As a result, in this scenario, the bottom strand would be considered the template strand. RNA polymerase would bind to the promoter region of the DNA, and then it will slide along the template strand, adding the complementary bases to make the RNA transcript. Note that in the RNA molecule, we would have the ribose as a base sugar for the nucleotides, whereas in DNA, we would have deoxyribose. Note that we now have a completed RNA transcript. This would be at the end of the termination phase. The genetic code, once again, exists in the presence of codons. These codons are present in the RNA strand, and they are important for coding for specific amino acids. As stated previously, the code consists of 64 different codons that code for 20 different amino acids. We now fortunately have an mRNA chart that can be used to predict what the amino acid would be from particular codons. This means that if we know what a gene sequence is, we can predict what the protein polypeptide chain would be. An important thing to note is that in using this chart, it is important that you understand that only the RNA molecule can be used. If you are given a DNA sequence, you would have to construct the RNA using the proper DNA template strand, and then you would have to do this prior to using the chart. Also, another important thing to understand is that you cannot use a tRNA molecule for this chart either. It is very important that you are only using the mRNA strand in this mRNA codon chart. If you use any other strand, you will not get the proper amino acid sequence. Another important thing to note is in using this chart, it is important to note where your start codon is located. This is very important in establishing the overall reading frame of your codons. If you do not determine where the proper start codon is, you will not establish the proper reading frame, which means that you will not get the correct polypeptide sequence. Thus, defining and locating your start codon is one of the most important steps in this process. If we take a look at the chart, which we will in the next slide, we will see that there is a start codon designated as AUG. We will also note that the start codon also codes for the amino acid methionine. This means that every single protein that is produced starts in the exact same way with methionine as the first amino acid in the polypeptide chain. Note that if you have additional start codons present in your sequence, this would simply designate that a methionine is inserted in the polypeptide sequence. It is important that you locate a start codon, but it is also important that you are locating the first start codon. This truly signifies the start of the protein. Any other start codon after that simply includes a methionine in the polypeptide sequence. The only way that it would start a new protein is if you had a stop codon present as well. It is a sequence of start and stops that will lead to the production of a secondary protein. Let's take a look at this chart. It is very important that you understand how these codon charts can be used. This is your typical codon chart. You can see from the diagram below that we have 3 different positions to consider. The first position in the codon, which is located at the 5' end of the codon, the second position, as well as the third position. The first position is located on the left of this box, or chart. The second position can be depicted by the bases listed at the top of the chart, and the third position is designated as the right of the chart. Note that we have 4 different possibilities for the first base of a codon. They can either be U, C, A, or G. By knowing the first base in your codon, you can determine which row you are dealing with in this chart. You would then go to consider the second base of a codon. This would then indicate what specific box you are dealing with within this chart. And finally, looking at the third codon position and what nucleotide base you have there, this will help you determine which specific codon you have and which specific amino acid this would code for. For instance, if we had a codon of, let's say, CUU, we would take a look at that codon and know that the first nucleotide in that sequence codon is C. This would indicate that along the left side of this chart, we would be dealing with all of the potential codons in the second row here that correspond to C as a first position. If we had U, we would then look to our top of our chart as a second mRNA base. Because this second position is U, we know that we are dealing with the first column of this chart. We now have the second row and first column to know that we are now looking at the one box that contains leucine. We know that in our codon we had CUU, so that means that U, or uracil, is in the third position. We can see within this chart that CUU would indeed code for leucine. As stated previously, there is only one way to start a protein and only one start codon, which corresponds to AUG, which also corresponds to the amino acid methionine. Once again, this is an important step in establishing the overall reading frame for your resultant protein. If we take a look at the chart, there were 3 different ways to end a protein. These are called your stop codons, which consist of UAA, UAG, and UGA. These are considered to be nonsense codons because they do not code for a specific amino acid in these instances. By determining your first start codon, AUG, the codons will then be read in the 5 to 3' direction. This corresponds to how the protein is built. The first amino acid, methionine, would be consistent with the position of the N-terminus on the polypeptide chain. Remember, the N-terminus refers to the fact that the amino group would be exposed. The other end on the polypeptide chain would be the C-terminus and would be consistent with the carboxyl group being exposed. This means that methionine is located at the N-terminus of all proteins and is the first amino acid in the sequence. The subsequent amino acids will be consistently added to the C-terminus as the polypeptide chain extends. Another important thing to understand about the genetic code is the fact that it is universal. This codon chart is the same and can be used for all organisms. There are very few exceptions to this rule. This provides very strong evidence of evolution from a common ancestor. This diagram here depicts the universal nature of the genetic code. Here we have a firefly gene